full. But let's get started. So I would like to welcome everyone to Theoretical and Computational Biophysics Group seminar series today. So it's my great pleasure and honor to host Professor Rommie Amaro from UCSD, University of California, San Diego, Department of Chemistry and Biochemistry, to the seminar today. And I'm going to introduce Rommie. But before that, please be reminded to turn off your microphone, keep your cameras on if you wish. We would like to actually keep it pretty informal, but still I would like to invite everybody to keep the questions for the end of the seminar, please. At which point I'm going to invite you to ask actually your questions in person and interact with the speaker. So with that, again, it's my pleasure to introduce Rommie. Rommie is one of us. So she was trained as a chemical engineer at University of Illinois Urbana-Champaign initially, and then she did her PhD in chemistry department, where she worked under Zan Schulten and worked on actually simulation of different systems, actually IgP synthase, and also continuing her work on his H system. And then she moved to the West Coast, doing a postdoctoral fellowship from NIH under Andrew McCammon at UCSD, and then had a detour to UC Irvine, joining the faculty there for a couple of years before she comes back to UCSD and joins the Department of Chemistry and Biochemistry, where she's an endowed chair and distinguished professor in the department. So Rommie is very active, and especially in social aspects of science. I have personally witnessed actually her involvement in a variety of activities. She is actually associated with quite a few editorial boards, and also a lot of societies and that are involved actually in engaging women and minorities in science and related aspects. Research-wise, Rommie has a strong emphasis on drug discovery elements of our business, molecular simulation, and also large-scale simulations that I'm sure many of you have heard of in terms of simulating large assemblies like capsids of viral systems, and her recent work on SARS-CoV-2 viral envelope simulation that I'm sure we're going to see some slides and some updates from her today during the seminar. Thank you very much, Rommie, for being with us and accepting our invitation to give us a talk, and we look forward to your seminar. Please.
Thank you, Ahmad. Well, as always, I'm so happy to be here, even virtually with all of you. It is a homecoming for me, of course, having spent so long in Urbana, and just really fantastic. And Ahmad, of course, working with Zan was really instrumental for me. Thank you, Zan. And of course, Ahmad too, you were the person who taught me really much of what I know about molecular dynamics at the beginning. You remember sitting next to me, teaching me how to protonate histidines correctly, even without servers. And this is the kind of stuff that we
Thank you.
do.
We have a paper together, I remember.
We do, a great one. Okay, so without further ado, and I will try to keep this to the proper time, but I'm gonna, of course, end up being long-winded because I have so many fun things to tell you about. So, it's been a crazy year. I counted in the past year, actually, I have given a lecture about SARS-CoV-2 computational microscopy 47 times in 52 weeks. So it's been crazy. And but I wanted to do something a little bit different today to give a talk to the home team in a way that sort of was a bit more personal reflection on the past year and just everything. So the title of my talk today is a little bit different. It's SARS-CoV-2 through the lens of Klaus's microscope, which I know all of us appreciate. And many of you, I'm sure, probably have already are aware of the work, or maybe you had a chance to see our work with SARS-CoV-2. It was featured in a beautiful article in the New York Times by Carl Zimmer in October. And that piece was in the science section, and it was just like phenomenal to see, science really being front and centerfold, our kind of science for the world. And one of the things, I think, so today I'm going to tell you sort of, the scientific story, the technical story behind these pictures, of course. But I also think it's been an interesting year helping to get computational biophysics and the type of things that we do, into the common vernacular for people. And I want to, for the world, so that people understand the value of what we as a community bring, and the kinds of science that we can learn. So I think that's something that I hope we all keep in mind. And, it reminds me of Klaus, because, he, of course, was a person for me and for many of us really of central importance, being a mentor, collaborator, a friend, sort of a kindred spirit. And he too had this ability to be, of course, amazingly technically rigorous. And I would challenge you to find someone, and Zan also, who could really sort of push people to really be their best, their level best. But at the same time, to be able to sort of, communicate the findings in a way that, anyone could understand. And that turns out to be something sort of very difficult, but that certainly we all took from him. But in any case, so there's this famous, I love this picture of Klaus. And then this quote from him about his goal being to look with mathematical and computational means at the inside of cells, one atom at a time, to decipher how living systems work. And that's what he strived for and never deflected from this goal. This is a famous quote. And anyway, I think the talk that I'll tell you about, I'll sort of go through the smaller pieces, but then it's going to culminate, of course, in sort of what we all did together. And there were many people who were part of this huge team to try to help carry on Klaus's legacy and to actually simulate the SARS-CoV-2 viral envelope with all of its many atoms. And, we were able to do it, I think, in part because many of us shared sort of the same inspiration from him. And I want to also, I love this picture too. This is a picture, Zan and Imad. Actually, Imad, I don't think you came with us on this trip, but this is a picture from Perth. So we had our second.
I took the picture.
Oh, you did. That's a shame. And Zan, we still have no pictures together. One from when you came, and from when you came to visit. We need more. But what I like about this picture, of course, I think I've said this before, is how long Klaus's arm looks, but it was just a way that he had of bringing people in, and I think making them feel included and valued. And certainly that was me. And I also want to comment that Elizabeth and Fatima and myself, all three of us, became academic professors, which is interesting because they're really still in our field is a dearth of women. And I think he and the center have in general done a very good job of of bringing up the next generation in a way that has been very diverse and successful. Okay, so all right, so I will breeze through this slide because I'm going to get really chatty on so many other slides where that I need to discuss. So all of us know, of course, I think the way that Klaus would speak about it, this method of molecular dynamic simulations as a computational microscope. And I personally, I love that analogy, of course, because it's very fitting. What we're doing is building these these integrated three-dimensional models of biological systems. And then we're, bringing them to life using numerical simulations and interrogating their structure and dynamics. And the important thing here is that we are doing this in a way that augments and extends where experimental biology can go. And that is, to me, that's the really exciting thing about these methods, is that we use them now in a way that they can prospectively tell us or predict what could be something interesting that we otherwise, would not have seen. So I tell you today about our work with the SARS-CoV-2 and some of the things that we've learned using this method. So, oh golly, you know, about a year ago, it was actually last February, was when things really sort of changed for so many of us in the world, of course, and also in the Amaro lab. So we had, that was a really important week. It was the week that actually we had finally published at long last our simulations of the influenza viral envelope. Again, here, this is where input from Klaus and his early support, you know, back starting in 2013 even was when we started building this system. And when the reviewers in the computational panels said that the work was too ambitious, Rommie, you need to wait for exascale computing to come, Klaus sort of really took me under his wing then, and he said, okay, you know, this happens. We all get rejected from proposals, but here, Rommie, I'm going to give you some of my discretionary allocation on, I guess the machine might have been Blue Waters at the time. I guess maybe around that time it had already come up. And that gave us the ability to prototype, of course, using NAMD and VMD at that scale, but to get that system up and running. And then that took 6 to 7 years actually to really build correctly, to rigorously analyze. And finally, anyway, at long last, we were able to publish that. That same week that this publication went to press, you know, something else in the world was happening, and that was that, you know, we started to really become more aware of COVID-19. And so what I'm showing here is the total confirmed COVID-19 cases on log scale for the early months of the pandemic from January through May. And that, you know, mid-February, you can see that the total number of cases in the United States was still very low. I think we had maybe less than a dozen known cases at that time, but we knew that the virus had already escaped mainland China, and it was actually looking very grim in the Lombardy region of Italy. And so this was when we started thinking, hey, we need to maybe do something with this virus. And then that same week, something else really important happened, and that was that Jason McClellan at the University of Texas, Austin, together with collaborators at the NIH, dropped into the bioarchive the first cryo-electron microscopy high resolution structure essentially of the SARS-CoV-2 spike. And when that data dropped, that was when we really pivoted our efforts towards SARS-CoV-2 in a very sort of real way. And that kicked off actually I can only describe it as an unimaginably insane timeline from a pace of research that encompassed a series of projects involving literally about 30 different collaborators all around the world, and 4 different and of the best supercomputing sites in the country, in order to build to this vision that was really in the spirit of Klaus, I have to say, to not only build the models and simulations of the single proteins, which I'll tell you about, these so-called spike proteins, but to really build it, to build the, you know, this sort of, this molecular simulation at scales, again, sort of breaking barriers and so forth. And so by October, as I'll tell you, we had been able to build and simulate this 305 million atom viral envelope simulation. So it was just a remarkable timeline of events. Okay, so let's talk a little bit about the science. So everybody here, I'm sure, is well aware of SARS-CoV-2, right? Here's a picture of the virus. Actually, I need to take this. I have to change my cursor back to an arrow because otherwise I can't play the movie. Okay, so, you know, it looks like the virus looks like a golf ball with spikes sticking out of it. It's a lipid enveloped virus. And those spikes are actually called spike proteins. And we care about the spike proteins so much because they sit on the outside of the virus. And so they're the first point of contact that the virus has with human cells. And so there are lots of people very interested in understanding the mechanisms of infection and so forth. The other reason we care about this protein is because it's also highly antigenic. And so it elicits or invokes a tremendous immune system response. And so in fact is part of all of the approved vaccines and plays a very important role. And hopefully some of you have already received your jabs that will allow us to, you know, eventually hopefully go back to something closer to what we knew before the pandemic hit. Okay, and so the majority of what we've studied really centers on the first step of infection. And what has to happen is that the spike protein, which I'm showing here on the left in sort of whitish gray, I'm going to talk about these little blue decorations quite a bit. These are glycans. I'll tell you more about those. But this spike protein has to interact with a molecule on the host cell or the human cell. And that's this molecule here in the center in yellow called ACE2. That's the angiotensin converting enzyme 2. And there's this light blue bit in the middle here, this receptor binding domain it's called, or the RBD. I'm going to keep coming back to that in the talk today, because this is really a very important part of the molecule. It's the part of the spike protein that has to make contact with ACE2 in order to precipitate infection. And you can see that very clearly here. And so this really important handshake is something that we've been trying to learn more about as we try to understand, sort of the business of the virus. So, as I mentioned, so we're interested in, of course, sort of in sort of developing models to learn about and simulations to learn about the spike protein. The spike protein is a trimer, and each protemer has, I'm showing here the sequence of the protemer. Each protemer has roughly almost 1,300 residues made up of a number of different domains. The bulk of the spike protein is constituted by the region that we call the head. And this is the structure shown here in the middle. This is the part of the spike protein that the cryo-electron microscopists can image to high resolution. And so this was, for example, Jason's structure and colleagues. This was published in Science in the middle of March. They gave us this relatively high resolution, the first relatively high resolution structure of the spike protein. You'll see that the little green bit here on the top, I'll keep coming back to that. That's that receptor binding domain. They had it resolved in what they call the up conformation, a 1-up conformation. Several weeks later, another fantastic structural biology group, the group of David Biesler at the University of Washington, published another study which largely recapitulated the structure of the 1-RBD upstate that Jason and his colleagues had also put forth, but they also presented sort of a modeled structure using symmetry of a closed spike structure, or where all of the RBDs were in the down conformation. And so, so the one issue though is that, or one challenge to structural biology in general is flexible bits of the proteins, right? And so this is really where efforts like NAMD molecular dynamics simulations and the high performance computing have a lot to give, because we can build back in these pieces of the structure that are otherwise not seen or missing in the experimental data, and then we can see what they actually look like when they move. So we modeled in the missing loops for both of these using modeler sort of standard approaches, and we were able to to build these complete models of these two systems. And so we start with this head, the head data that comes from the structural biologists, and then we needed to build a full structure. We also had to fill in some of the other missing pieces. In addition to flexible loops that were missing, the head is connected. The head of the spike protein is connected to the viral membrane through this part called the stalk, which you can see here. And this is a transmembrane domain. And so this is a very relatively simple trimeric coil-coil helical bundle. We use modeler to make that also. And then there's this bit that sticks down inside of the virus that's called the cytoplasmic tail, or the intravirion domain. And we modeled that also. And so this is what our sort of full end-to-end structure looks like, with all of the bits actually built in. So then the other thing that the structural biologists have trouble seeing are N and O linked glycans. And so, so probably most of you know that glycans are a really important post-translational modification. They carry out all sorts of interesting biological phenomena, and including helping the proteins fold and so forth. But for viruses, they have a special role. And that is that viruses have evolved the ability to essentially decorate themselves in these sugar-like molecules in order to look like our human cells, which are also decorated in these sugar-like molecules. And so by doing that, they sort of effectively can disguise themselves and their foreign proteins from our human immune system. And so, with SARS-CoV-2, it was, we started building the structures and working with them. And then in I guess about April maybe, and Lorenzo and others in my team would remember probably the date to be exact because we had to rebuild the darn thing. But they actually came forth with a very nice glycomic study. This is a group of Max Crispin again together with Jason to look at a site-specific, he gave us basically using glycomics, the site-specific glycan composition, at each of these places on the spike protein. And so with this data, and also there were other groups who came forth with supporting data also, for example, Paris Duazadi at the University of Georgia, where she also found some O-linked glycans. With this data, we were able to rebuild those missing bits from the, that were missing in the structure, but we know that are there biologically. So just to give you a better idea of what this looks like, so there are on each of those trimers that I was telling you about, there's 22 N-linked glycans and 2 O-linked glycans. Each of these are encoded at a particular asparagine residue. We call these sequons, and we know from the glycomics that they're of a particular type. And so these sugars, they look like this. They're these like branched chains of sugars with different compositions, and they have very specific linkages. And so what I try to impress on people is sort of this was the hard work and the sleepless nights of Lorenzo Castellino and Zied Gayeb in particular in my group, who really at the early days of the pandemic, really trying to get all of the residues in the spike protein correct, including in the missing loops where there were missing disulfide bridges. You have all of the complexity of the nearly 4,000 residues of the protein. And then on top of that, you have this gift of the complexity of the glycans. And so there were many, it's nerve-wracking, of course, as you know. And as Klaus said, I should say this now because I'll forget it later. He used to say, he'd say, small systems, sort of small problems, or, small systems, you have a few problems, but with big systems, there's still little problems, but you have millions of them. You have so many more. And so I think we've sort of felt that pain point here. Okay, so then beyond the glycans, the other thing that we wanted to do, we needed to build a model of the membrane. And we know that the virus buds off of the endoplasmic reticulum in the Golgi intermediate compartment. And thanks to many years of basic biological research, we know what the different lipid compositions for each of these organelles and compartments are. So we basically, reconstituted an in silico membrane with this rough molecular stoichiometry. We did have cholesterol in there, as we were belaboring or bemoaning. Ahmad, cholesterol is such a just pain in the butt molecule, if I'll say it myself, because it has that darn ring, and the other lipids, they love to pierce that ring. Yes, we did experience that pain, but we made it through anyway. So this is basically what the composition was. Now, there's one more part I want to tell you about. So here we're looking at the bottom of the spike protein. Across the middle of the screen, you see the lipid membrane. Those different phospholipids I was telling you about on the previous slide, you can see them colored here in different shades of pink and orange and yellow. But there's one more bit. So the cytoplasmic tail is littered with cysteine residues. And on these cysteine residues is yet another post-translational modification. These guys, many of these cysteines are palmitoylated. And you can see them here. These are these little shapes that look like caterpillars. These are basically just greasy, aliphatic carbon, molecules, carbonaceous molecules that basically they snorkel up into the membrane and help anchor the spike protein in the viral membrane. So our model had that too. And so this is what the full end-to-end spike structure actually looked like. So just to recap, we have all of the parts that should be there. So a mix of computational modeling plus experiment. We had built different system types. So we had we used the data from the RBD and the up conformation, as well as that closed structure from Fiesler. We have all of the glycans sort of running along the whole protein. You can see how many there are. We have the viral membrane, the palmitoylated cysteines, and this is what the fully built structure looks like at the end of the day. This is about 1.7 million atoms or so. And then we invoke the miracle of NAMD, right? So here, where we were using generally the Charm 36 force field, and we were running NAMD2 on TAC Frontera, we were able to get about 60 nanoseconds per day on 256 nodes, which was pretty darn good, we thought. And thanks to the generous use of TAC, we were able to generate about 4 microseconds of data for each of the RBD up and down conformations. And here I want to echo Zan Schulten, because I think it's really important that we always remind ourselves, but also other people, why we do this is because this is, molecular dynamic simulations are numerical statistical mechanics, right? So it's not just a pretty picture. It's not just an animated movie or video game, right? These computations are done in accordance with the laws of statistical mechanics. And so we can, at the end of the day, extract back out from averaged microscopic properties, experimentally testable macroscopic observables. And so this means that we can connect our data sets exactly close to, not exactly, but we can make connections with things like binding free energies, heat capacities, actually things that can be tested experimentally. And this is the real power of these simulations. And this is why we pay all the price and make, to run these simulations. And we go through all the pain that we do. Okay, so what did we learn? So part of the reason why this study was so exciting and so well received was because we showed the world for the first time what the spike protein, the full length spike protein actually looked like. So on the left is a spike protein. I'm showing it in light blue surface representation, viral membranes down at the bottom. And this is essentially what the structural biologists see. They see the protein. They can see, to be clear, they can very often see sort of the first stub of the glycan, in this case, the glycan unit, the first unit, but there's so much of the sugar they can't see. On the right is actually what the spike protein is a better picture, a more accurate picture of what the spike protein actually looks like. The blue fuzzy bits are the glycans. And so each of these sort of blue tough fuzz balls that you see basically represents a composite image of many snapshots of a single glycan taken over the course of 1 microsecond of dynamics, which is really not that long, but it gives us a sense for, it gives you a sense for the volume of space that is swept out by each of these different glycans. And so you can imagine you have this fast motion, dynamical motion of these glycans. And they basically, that's what creates that disguise, that dynamic disguise or that shield from the immune system. So it's just was really cool to see. And people care about this information. It's not only cool to know, well, where is the shield, but also where it's not, because the shield is sort of like a cloak of armor, if you will, in some sense, for the spike protein. And so places where there are holes or chinks in the armor, if you will, these are vulnerabilities. People consider these to be vulnerabilities, both for neutralizing antibody binding as well as potentially for the development of small molecule drugs. So lots of folks interested in these data sets. Okay, another cool thing was that the simulations actually showed us why the spike was found in these 2 different conformations. So why it actually opens and closes. So here I'm showing on the left is a view of the closed spike. So the all RBD down spike. This is looking down from the top. So this is sort of like if you were ACE2, what you would see. And that receptor binding domain that I mentioned, I always try to show that in the light blue, you can see how it's mostly shielded. It's a large part of it is shielded. From the side, you can see how that blue bit is really sort of tucked down beneath that sugar shield. And this is in contrast to the open or the 1 RBD, the RBD up state of the spike. So you can see here in the open state how the spike presents a completely different binding interface for the RBD. And this is from the top view and then also from the side view. And it's just so important because it is this part right here that has to make that handshake with ACE2. And so you can see, so I like to think of this, that if the structural biologists could have seen the glycans, would they have called it up and down? I think they might have called it exposed or shielded, or defending mode and attack mode. But so this is the type of insights, the type of things that we can learn from the simulations. And of course, now we have these huge data sets, relatively huge data sets that can be mined for all sorts of interesting things. We of course look at things like accessible surface area, and we can characterize shielding, glycan shielding and protection. There are a lot of people who are interested in trying to understand where neutralizing antibodies are binding and where they're not binding, right? Because this is, of course, of huge interest therapeutically. So many, I'll just show, this is a relatively old picture. At the time when we were making these studies, I have to say this, the whole, working in this area, for those of you also who have been working in this area, it has been in some ways like a blessing and a curse there's been some 60,000 preprints in about a year's time. And so as more data keeps flying at you as you're analyzing your own data. So I'll just say this is a relatively older image, but trying to characterize some of the first discovered neutralizing antibodies. Since then, there's now hundreds of structures, but this sort of captures the first handful, where we knew at that time that many of the neutralizing antibodies were in fact binding to that up RBD. They recognize that same spot that the spike protein uses to bind ACE2. And so blocking that is one way, in fact, that it can curb infection. But there are other interesting features, other interesting neutralizing antibodies, like this cryptic antibody CR3022 that was discovered by Ian Wilson. One of the things interesting about that one, which is highlighted in green, is that actually it looks like the antibodies can only bind there if there are 2 RBDs up. Then there are a whole bunch of other antibodies. At the time, this was the only one, 4A8, which actually don't bind the receptor binding domain, but they bind the N-terminal domain, which is like sort of sitting next to it on the top of the spike. And it turns out we now know that the N-terminal domain is a super antigenic part of the molecule and almost a place where many neutralizing antibodies bind. But so you can see that there's many, as I mentioned in the beginning, it's a highly antigenic molecule. And so your immune response is polyclonal, which is also really good. That means that you're making multiple different types of antibodies against any single spike, including in other completely different regions. Like, for example, this one down here, this is 1A9 binding down in the connecting domain. And one of the interesting things about this is you can see, or you can kind of see, it's pretty much occluded, how the connecting domain antibody, and that's sort of like in this region of the spike that's literally at the bottom of the head near where the spike head connects to the stalk. But that's actually sort of in a buried region. And here I also want to just mention that these initial studies just looked at the low-lying sort of short time scale dynamics around this closed and one up RBD sort of pre-fusion state. But we know that this molecule is, and I don't really do it justice, but this spike protein is it's a spring-loaded molecular machine that goes through a huge conformational transition as it facilitates host cell and viral fusion. And so we're just focusing on just this tiny part, of its sort of biological events that it carries out. But this reminds us of that because we can't really rationalize the binding of that antibody unless we think about the other states that this molecule actually populates. Okay, so beyond shielding, there was something really interesting that came out. And this has been a wonderful collaboration with Elisa Fada. She is a terrific glycobiologist / glycochemist at Maynooth University. And we found something really interesting. And that was that when we were looking at the closed state of the spike, one thing that jumped out at us was that there was one glycan shown here in blue and green that here we're looking again top down on the spike, that it always sort of seemed to stick outwards in solution. But when we looked at the open structure, or that one RBD up, what we noticed was something different. And that was that 2 of these guys were sticking out, but one of them actually in the N-terminal domain that's adjacent or just next to the one RBD up, it actually was pointing inwards. And so we took a closer look. And here what I'm showing you is the top of the spike protein again. I'm showing in red and gray and blue the different domains of the spike protein in surface representation. This is that RBD, trying to keep that in blue. These 2 glycans, so the dark blue bit is the part of the glycan that the cryo-electron microscopists could see. They see that first sugar unit. So when we rebuilt the rest of the side chains, of the actual glycans, what we saw was that they so perfectly filled the void space that was created when the RBD actually went into the up conformation. And so this led us to suggest that we decided, well, what it looks like, it looks like these guys are propping it up like a kickstand on a bicycle. So what would happen if we mutate these asparagines to alanine? Okay, and when we do that, you can't add a glycan there. So our hypothesis was that if we made this mutation, this receptor binding domain would collapse down. It's no longer structurally supported. And so we ran a whole bunch more molecular dynamic simulations of this in silico mutant. And what we saw was the collapse, essentially, of that state, actually at rather short time scales. So, on the microseconds time scale of just ordinary dynamics. So at that point, I contacted Jason McClelland, and he is, as I mentioned already, he was the person who put forth the first structure of the SARS-CoV-2 spike. He's co-inventor on the Moderna vaccine, and famously now, perhaps Nobel Prize famously one day, he was among the people who really, I think, the team lead who created the so-called 2P mutation. And so I'm probably going to run out of time. Yeah, I'm not going to be able to tell you too much about that. But this turns out to be a really, really important mutation to this spike, which we can talk about later. But so this was our sort of wild type positive control. And then he also created a version of the spike that actually has this disulfide linkage so that it's always in the down conformation. So this is our negative control. These are biolayer interferometry experiments that his group set up. So this is Jason and Jory and Christy. And they set these up to actually look at the point mutations and to see then what would be the effect, the biological effect in terms of the spike's ability to interact with ACE2. And what this study showed us was that these mutations actually did reduce the ability of the spike to interact with ACE2, which you supported our hypothesis that actually that these glycans were actually helping to stabilize the spike in the up conformation. And so this work was published in ACDS Central Science in October. And, I'll just sort of recap that in addition to sort of showing the shielding, beyond shielding, these simulations established a structural role for glycans. And what we didn't really even appreciate at that time was that actually glycans, this was the first time that glycans had been shown for any protein to actually play a role like in the actual mechanism, for example, of infection. And so that has been, I would say, very well received and very cool that we use simulation to show and extend what we know about glycans in general, not only specific to SARS-CoV-2. So that was really cool. Okay, then beyond that, so then, we wanted to actually see if we can do something more. And so as all of us know, I mean, one of the big challenges for molecular dynamic simulation, as beautiful as it is, I love to see all the atoms wiggling and jiggling around. We pay a price for that. And that's, we have this challenge of timescales. And so we wanted to see if we could simulate the full transition of the closed to the open structure. So this functional transition of, what does it look like when it goes from down to up? But we knew it was not going to be possible to do this with conventional molecular dynamics because it's just it's like big sampling problem. So here we partnered with Lillian Chong, who is a wonderful, fantastic, brilliant methods developer and scientist at the University of Pittsburgh. And she's been developing for some years this weighted ensemble MD technique. She actually spoke in the series, I think maybe a few weeks ago, or she's speaking soon in this series. But the idea is, of course, that in sort of conventional MD, we spend a lot of time sort of waiting for something interesting to happen. We're sampling a lot in these sort of wells in the energy landscape, and her method is one that sort of helps to enrich the probability of success for these simulations to actually go over these interesting barriers. And so we use that. It's her program called WESPA. Weighted ensemble simulation with parallelization and analysis, which is actually coded in Amber MD. And here we used a different machine at TAC. We took over their Longhorn GPU farm, essentially. We took over 100 NVIDIA V100 GPUs for about 2 or 3 weeks. And we were able to, using her method, generate, actually, this is an older slide. Oh, darn. We actually were able to generate, I'll just speak over this slide, about 300 independent opening pathways. And that took about 23 days. This was in contrast to if we wanted to use conventional molecular dynamics, we would need to, if we wanted to just sample one of those, knowing what we now know from real-time conformational sampling using FRET experiments, which was a beautiful study that I unfortunately don't have on the slide anymore because this is accidentally old slide I inserted. It was published by the group of Walter Mothis from, I believe, at Yale in Cell Host Microbe in 2020. They think that the opening and closing motion of the spike happens on the order of seconds. So if we use that, if we know that, then basically it would take us about 800 years or something ridiculous to use conventional MD and sort of hope to get even one conformational sort of transition to the up state. So this is a way, this is just another way of saying this is it's a very efficient way of sampling these interesting functional transitions in biomolecules. And so I hope this movie plays. I knew it was going to do that, but that's okay. I have many more movies. But so basically what I would have liked to show you, but the movie failed, is that we start with this closed structure and we are simulating and basically using her method, we're able to sample that opening conformation. And what's interesting about this is that residues, for example, so I'm showing here all the glycans like I showed you before in dark blue, but this one guy here in pink, this is a glycan that if we just looked at the, I'll just talk to this slide even though the movie's not playing. If we just looked at, from like in our initial simulations, if we just looked at the end states, so the closed state and the open state, this glycan here doesn't look to be doing anything interesting. But when we look at the opening pathway, you know, when we're able to actually simulate the pathway, then you see very clearly, and I'll show you hopefully in 2 more slides, that this residue actually is doing something really interesting. It's performing a role, a gating role. But so one of the nice things that we can do with this type of data is we can look at interactions all up and down along the opening pathway, right? Because we get these, we're able to generate 300 different independent opening trajectories essentially. And then we do like an ensemble sort of analysis of those trajectories. And so starting with the down state, we can then sort of characterize along this sort of pathway from the down to like an intermediate or transient state to the up state. We can sort of see what types of interactions are changing along the way, which is really, you know, informative in terms of trying to understand mechanism, right? And then also one of the interesting things was our simulations, we just let them keep running. And so it got to the up state, but then we also saw that it just kept opening. And that was really cool too. So we sampled these sort of wide open states, which is neat because it starts to give us clues about what the pathways might look like en route to S1 shedding. Because, you know, the spike protein actually, as I sort of mentioned, but I don't do it justice here because I don't show you like the whole pathway of it. But the whole top part of the spike protein essentially peels off. And then you have these central helices that polymerize sort of like up into and connect with the host cell membrane. So it's just like really amazing sort of structural cascade or sequence of events. So anyway, so we start to get some glimpses of like going further along in that process. Okay, and hopefully this movie plays. So this is, again, the top of the spike protein. What I'm showing is the RBD, again, in blue, but now as sort of like, you know, cartoon representation. And here we have this glycan that we all of a sudden care a lot about, and we're watching it. And what we see is that this glycan basically sort of does like a hand jive motion with different residues on the RBD, and then eventually helps almost kick up and out that RBD into that up state, an open state. So from this, you know, and again, I just want to emphasize that when we just looked at these, the 2 end states of open and closed, that N343 kind of just does not look that interesting. But again, when we actually looked at the pathway, we see that it has, in fact, an important role to play. And what we found was that it's actually, first, it's sort of shielding the spike protein, then it helps kick it out, or that RBD. And so we proposed that it's actually a glycan, it's acting like a gate. And so we went back to Jason, who was kind enough to go back to his biolayer interferometry experiments, and they ran a whole bunch of different mutations, not only of N343, as I'm showing here, but some other residues that we thought were making, you know, interesting hydrogen bond and other interactions. And we, you know, see this reduction again in the ability of the ACE2, the spike to interact with ACE2. And so again, that sort of corroborated our hypotheses, and this paper is nearing to get impressed, but it's been into the bioarchive now. Okay, and then beyond that, of course, you know, we want to understand how is this thing actually, how is the spike protein actually interacting with the ACE2 molecule. So we have even more simulations of actually ACE2 in the viral membrane, in the human membrane, the host cell membrane. And so here, one of the interesting things we found was that ACE2 actually has a really remarkable degree of flexibility, and it can almost lay down and sort of touch the host cell membrane. And we sort of, you know, because it's interesting, because when you see the cryo-EM structures, and people tend to think about it just sort of standing very much like a soldier kind of sticking up, but it actually has this huge amount of wiggle room. And so we think that that sort of that ability to bend and so forth is actually potentially playing a role in sort of the mechanics of infection and helping to facilitate that. And then hopefully this movie will play. Okay, so this is this will combine several movies. So here is the movie that I wanted to show you of using weighted ensemble to go from the RBD down or closed state to the up or open state. And so here, you know, we're sort of sampling all these dynamics, and then eventually you're going to see the RBD open. There it goes. And then once it's open, we have combined it now, simulations of the sort of across a parallel two membrane system where we have ACE2 in the host membrane and the spike in the viral membrane. And so this is cool because it allows us to look at the interactions across the interface, but also because it lets us look at all these other degrees of freedom and behaviors that are happening. And here we saw like a lot of stalk bending, and I don't again do it justice here because I need to wrap this talk up. But we have been collaborating with Shantanu Jha and Arvind Ramanathan. Shantanu's at Rutgers and Arvind is at Argonne National Labs. And they have a method called Deep Drive MD, which I really should put a formal slide in here to show. And Anda also has been working on this a lot. She's part, you know, I believe part of the center too. Here it's a way to basically help sort of improve in some sense the conformational sampling by sort of nudging the system to go into regions where interesting things are happening. And so this is something we're working to, it was part of our Gordon Bell paper, which I'm about to transition to talk about, but it's also something we're working to write up more comprehensively. So what does that mean? So what we saw was that we were able to use Deep Drive MD to help essentially accelerate in some sense or sample more effectively this stalk bending. And so where we basically identified different modes of motion and then chose restarts basically in these sort of more interesting places on the map, if you will. And so you can see sort of these are sort of histograms of angles that we've sampled in these ankle, knee, and hip type joint regions, of this spike stalk, and how in the first reps we have some sort of distribution. This is indicated for all of these in sort of like the darker colors, and then in the lighter colors would be when we were able to do this restart that iterated with this Deep Drive MD. I didn't explain that very well. It probably needs a whole talk to itself, but I just wanted to make sure I mentioned it. Okay, and then you know, then part of the vision that we had was to really to see if we could sort of redo what we had done with the flu, but of course now with SARS-CoV-2. And so, you know, to simulate these viruses, I think the single biggest obstacle is really getting the lipids correct. That viral envelope, the lipid envelope, is really a huge challenge there, because you know, you have differences in the curvature of the inner and outer leaflets, causes all sorts of interesting problems to happen. And, you know, I know that you at the center have been working on this challenge for many years. And, you know, we've also tried to help develop tools and so forth like LipidRepper to address these challenges too. But here, basically what's required is an iterative, intensive sort of remodeling process where, you know, we're finding these little holes and nits and filling them up and then trying to insert the spike proteins and so on and so forth. And so it's this really, it's quite an intensive process. And this was done by mainly by Abigail Domer in my group, and she did just a fantastic job getting this up and running in time to hit the Gordon Bell. So, you know, at some point along the way, they released that they were going to, ACM was going to have a special Gordon Bell Prize for COVID-19. And I have to say, I think I was in the group when Klaus and Laximant Calais and your teams won that Gordon Bell. And I remember what a big deal that was. And when they announced it, I thought, you know, maybe we have a chance. Maybe we could sort of repeat what, in some sense, what Klaus had won some years ago. I guess it might have been 2002 or 2003 when I was a grad student. And so that was there. And then we started talking about it, and we assembled this team together with John and Jim and Anda and Arvin and, oh my gosh, so many people, 30 people. I can't even name everyone. And then came, oh my God, now if we're really going to do this, we got to do this. And so I'm showing this picture. This is actually a picture from Perth. Zan, you didn't take this picture. This picture was taken by Elizabeth because it was 3:35 in the morning, the day before we were going to do this, our traveling roadshow. And I'm laughing because we are so jet lagged and so exhausted. And we're trying to like get all these laptops to like load all the software for all of those darn tutorials. And I know all of you at the center, you've lived through this, but you know, when we were first, this was our first roadshow. You know, we had done it in UIUC, and then we mounted it onto these laptops. And, but I put this here because this reminded me so much of, you know, when we were going through this process with the Gordon Bell, it was this excitement of, are we really going to make this? You know, we really didn't know if we were going to make it. We were trying to squish so much in, and we were working in the night, like through the night. I mean, we did not stop, but we were able to, you know, it was just around the clock efforts from so many people that got it done. And in the end, this is just a little bit of a wiggling and jiggling showing that we did it. You know, we were able to get the full viral envelope using the cryo-electron tomography data now, where they have the correct patterning and density of the spike proteins. So integrating the single particle data with tomography data to build the envelope, you know, this structure, and actually get it up and running so that it flies, you know, on this was actually, we had prototype runs done on Frontera and then ported them over to Summit to do the performance benchmarks. And I'll just tell you that this was really what I call the Gordon Bell timeline of insanity, because you know, we started really thinking, okay, we're going to try to do this maybe in like July, and we were working, everybody was working on all their different pieces, because it was the individual spike, it was the weighted ensemble, it was the deep drive MD, and it was this big virus. And so we started doing these prototyping runs and actual science runs starting in August. But the due date was October 9th. I will tell you, we did not even start writing that paper until October 7th. That was the first time that we started to really put words in it. The virion, the 300 million thing, had not even completed yet. The paper, yeah, so it was due October 9th at any time or any place in the world, which meant that was about, I guess it's like, it's in like French Polynesia. So it was like we had till about 3 a.m. Pacific time. And it was just, I tried to go back and go to our Slack channel, folks, because there's 20 people on the Slack channel, many of them I'm sure who are at this talk. Unfortunately, we had so many messages and I don't have like the paid for Slack version, so I can't go back to them, but it was just round the clock, pushing. The 305 million atom virion simulation finished running at 6 p.m. on the date of the paper submission due date. So at 6 hours before we actually hit submit, we actually got the performance benchmarks for the thing. That's how close it all was. And you know, this eventually we had actually submitted with 3 hours to spare, which I thought was pretty impressive. But then, which was just fully Klaus, I'm sorry, then it wasn't, the paper wasn't enough. Then it was like the revenge of the site visit, because not only did we have the paper, but then we had to give a presentation about it. So it really felt just like a site visit. And I think everyone was sort of channeling that same energy of just, you know, leaving it all on the field. And so so we submitted the paper, and then we had about a month to create this presentation. And I will say that, but what I think made it even worse, and I was thinking about if Klaus was here, if you all, and maybe you all, no, you wouldn't have experienced it, I don't think, but imagine that you had to, so we had to pre-record our Gordon Bell presentation. And I have to say, like, at first, I think all of us, many of you know the pain of this. At first you think, oh, pre-recording, this is going to be great because I don't have like the nerves of like the live show. But then you realize that it's like so much more work to actually record something, because now you can control every slide, every word, every audio recording bass versus, all of the audio and the transitions. It's like it was like perfecting the site visits, but like honestly, I felt like it was almost on steroids. And it was really the hard work of so many folks, and we just absolutely left everything on the field. And so this was the group of everybody coming together. I can't say all the names. There's 30 people, basically 30 authors, and many of you in Urbana. Was so fantastic, and just 4 different computing sites. It was really something special. And then we did have the privilege to to actually win the Gordon Bell. That was you never expect for that sort of stuff to happen, but it was really cool that it did. We enjoyed that very much. Yeah, and then I'll just say, so the timeline was sort of like the paper submission was October 9th. It was like, whew, we made it. And then we were all thinking, okay, now we're going to sleep. But then the New York Times released that really nice Zimmer article. I was thinking, like, I just want to get off Twitter and relax. And then that same morning, they released that, and it was just like explosion of, more stuff. Then the work to the presentation, the win. And I have to say, the recovery, it did also feel like the site visits, where it's like there's just such a feeling of afterwards that I think I finally recovered. I think it took at least 2 or 3 months to actually feel like I could do something else again. But anyway, then in the closing part, I'm sorry I've gone a little bit long. I'm trying to leave time for discussion. I just want to say this whole thing with the virus, there's been so much death and devastation. It's hard to even really pretend to think about having a silver lining, but still we try to find them. And I think that I was really sort of heartened by the response of our community, I have to say. Early on in the pandemic, back when we were trying to build those perfect glycoprofiles on this terribly complicated spike protein and get all the disulfide bridges and protonation states correct, and the pain of all that, we realized that we had to do something different. It would make absolutely no sense for every group in the world to like redo that. Why won't we share with each other the models that we made, the knowledge that we have in advance of publication? And so it was such a remarkable response from the community. Over 200 different simulation groups helped to develop a shared set of principles around open science in this space. And I like to hold this up because I think that it's important for our field to share more in general, and it showed us that we can do it. It also led to the development of a wonderful data sharing site at the COVID, this Molecular Sciences Software Institute quickly propped up a data sharing site for all of our trajectories and simulation files and so forth. And so those Frontera data sets that we generated have been shared at over 4,000 times. They've been pulled down and looked at people all over the world, and other people are depositing in here too, right? So this is just a wonderful new mode, I hope, that we carry forth with us as we continue, not just in SARS-CoV-2, but in other areas that our field works. And it's super important to emphasize and continue to reiterate, especially in the funding environments, how critical it is that we have high quality community software that's open access and that does everything that the end-to-end scientist needs to get their experiment done, right? I mean, because of NAMD and because of VMD and the decades of investment by the NIH and the decades of the hard work of your team, it's a tool that so many people could run with, and I don't know, probably you guys have done the analysis, but I would love to see it. How much new knowledge, even in the space of SARS-CoV-2, was gained from your tools. I'm sure it was enormous. Anyway, so it's really important that we have those types of things available. And I also just wanted to say, that the COVID-19 HPC consortium was also really key. Absolutely would have been unimaginable to think that we could have not only built the virus, but gotten the compute cycles to run that thing. Because I'll tell you, for the flu, we tried, it took 2 to 3 years to convince the panels to give us the time to run the MD. And here it was like, we had it. And they said, just do it. And they opened it up. And it was like, when they did that, all that we could accomplish, it was just so cool. So this was really important because it took what ordinarily, I mean, not just for the big virus, which would have literally been unimaginable. But like for even the smaller systems, like the spike and the proteases and so forth, you know, they made compute cycles available for researchers like us, you know, on the order of weeks, which you just like, is unheard of, as opposed to ordinarily being 6 to 8 months to a year to get the time. Okay. So many people to thank, of course, because this was like a huge team, and as I sort of already have, but I'll just give shout outs special for the spike protein in particular to Lorenzo Castellino, Ziad Gayeb, who's left the group by now, Abigail Dahmer, Tara Stain, Shirley Ahn. They really pioneered the weighted ensemble work together with Lillian Chong. There's just so many people. I'm not going to be able to say all the names, but I'm so grateful for all of their work, and especially Elisa Fada at Maynooth University for the glycan expertise, and Jason McClellan for being just a fantastic collaborator, and the funding agencies, not only for the personnel support, but also for compute support. And you know, one more thing I'm going to say, because I have the floor, is I was thinking it was just about 5 years ago today, almost to the day, that I was in Beckman, and I was giving a talk. TCBG had invited me for that talk, and I was so happy to come back and you know, and just share with all of you sort of what we had been up to, you know, as an independent PI, what I had been up to. And I gave the talk, and I remember Klaus was there, and he was very thin at that time, but he was there, and he said, he asked me, he's just like, you know, Romy, why do you build and you simulate these ever larger systems? And I was standing up there, and I you know, I said, oh, because of this and because of that and because of this and because of that. And then he said, at the end of my rambling, he basically said, no, you know, you do it because you can and because we can. And I have to say, I do agree with that. And it's something that, you know, always sticks with me, you know, this idea that we, you know, you don't necessarily sort of need a reason to climb a mountain other than to see what the view is going to be and to sort of know what can be accomplished. And that was certainly this, you know, the spirit of Klaus. And so much, I say and on the bottom, because so much more follows from that. You know, you have these achievements that break new ground, and then from there, you set the stage for so much else, you know, to advance. And I'm going to end with this. I can leave this slide up for the Q&A if you want or not, but this was during a wonderful time in grad school. This is Elizabeth Villa, who I'm sure has logged off by now because she has so much stuff to do. There's Imad, Klaus, and Zan, and Jordy, and myself. Oh, you're here. A wonderful trip to San Francisco, another traveling road show, but after we had worked out most of the bugs, so we had time to bike and relax. But it's just a famous, one of my favorite shots of the group. So with that, I'm sorry for going a little long, and I'm happy to answer all types of questions or just to chat.
Thank you very much, Romy, for the inspiring talk and really beautiful example of how much we can learn from simulating and modeling these very large systems. Actually, it was really impressive. Thanks for sharing us your story and sometimes your frustration with funding agencies. We share that with you. I mean, it's unbelievable how skeptic they still are in terms of simulating these large systems. We have had similar experiences with them as well. So I'm sure there are lots of questions. So if you have any questions, please raise your hand or type them in the chat box. I'm going to invite you to ask your question yourself. I'm going to start myself sort of talking about these large systems. We spend a lot of time putting together the membrane. You and my group also several years for different systems. Did you see any effect of the membranes on the spike protein or any interesting interaction, observation from this model?
I wish that I could say yes to that, because we have put in the pain to make it and then, you know, do it all. We haven't gotten to really analyze anything yet. So it's too soon for us to say. But I am encouraged that there are a lot of groups who are really interested, of course, in sort of in looking more closely at the interactions of the membrane with the spike and the E proteins and all sorts of other, you know, fatty acids that might be in the system. So it's something that unfortunately we haven't dug in on yet. But also it's a good thing why I'm happy to share that. I'm happy that hopeful one day we're going to share all those big data sets too, so that others can look to see and mine them for interesting things.
Absolutely, fair enough. Zan?
Yeah, Romy, that was just a beautiful talk. And love the picture. Say there is one with us.
Yes, we do. We do. We're always, yes, we do.
Right. So, but a quick, two things came up as I was listening, particularly about your mutation study, is of course the mutants that we're all worried about. And some of the pathways came enticingly close to those ones that are on everybody's mind, like 484, 417, and 501. And I just can't imagine you haven't also looked at those. And
oh, sure. Yeah. I couldn't tell you about everything we've done. I had to be, I just basically cut out all the stuff that was in the Gordon Bell, but we, and so the mutations came after that. So yeah, we've studied a number of them. And actually now that I still have my slide up, here's one. I took this out to make room for all the other stuff. So yeah, so, you know, these mutations are really interesting, and these methods are very good, I think, at giving rapid assessment of these variants of concerns in terms of what are the mechanisms of what are they doing, right? So 484 is right at that RBD interface, and it's switching the charge on the residue. And so that's obviously, you know, hampering the ability to interact with certain neutralizing antibodies. The N501Y, you know, makes additional stacking interactions and so forth. We can look at that. This study here, which has been in the archive since January, I guess, is one where they lab passaged a virus in a potent polyclonal patient sera. And when they did that, after about 90 days, it did all sorts of stuff to try to escape the neutralizing antibodies. And one interesting thing that it did was in the N-terminal domain, adopted an 11 amino acid loop insertion, which is extreme loop remodeling, which basically kicks off every NTD neutralizing antibody that's there, but also introduced a new glycan sequence. And so here we've just modeled, again, using the tools of NAMD and VMD, what that would do to the interaction of the N-terminal domain with the neutralizing antibody, which I'm showing here, with a potential neutralizing antibody, which we're showing in gray. And you can see that if we rebuild the glycan here, which is in dark blue, how it just basically would obliterate the ability of anything to bind at that site. So yeah, we've looked at that. And of course, there's tons of interest. And it's just, we're trying to get some sleep too.
Question about your glycans, because this was really impressive. So as you say, it's sort of a trick to, you know, trick our own immune system to think we're like one of our proteins, but everybody has a slightly different set of glycans.
Oh, it's such a good question. Yes.
So, you know, there was a story a couple months ago, maybe there's a correlation with blood type, which also involves glycosylation and your survival with these things. So,
yes.
Any further comments on that front, by the way?
Just we are working on it. We are trying to work on that with some of the best experimentalists in the world, and it just takes time. So yeah, there's something to it. I mean, the glycans are the whole, yeah. I don't have anything concrete to say yet, other than we're looking at it with similar tools and trying to disentangle some of what they sort of, you know about that.
Well, that's the next level question. That's great. A beautiful work.
Thank you.
Absolutely. Okay, so then let's go to students. Kevin, you have a question?
Yes. Hi, Romy. It's nice seeing you again.
Hi. Wait, which Kevin is this? I don't see your face.
Kevin Chang.
Oh.
It's okay. Kevin Chang. Okay, yeah.
Good. Wonderful talk. But I was wondering if, based on your results, do you think there's a strong correlation between the flexibility of the stock and the infectivity of the virus?
A strong correlation. So that is something that has not been experimentally vetted, I think. So, I mean, our hypotheses, and I'm sure other people would probably agree that stock is there and it bends for a reason. It's probably helping to, you know, make the interactions in more promiscuous ways. You know, it probably gives some advantage to the virus in terms of finding and make, you know, the contact with ACE2 and sort of getting the cells together for fusion. But it hasn't been experimentally tested yet. But that is something that at least we're hoping maybe to get to, actually, with the work with ANDA. But we don't have it done yet. And maybe
Oh.
there are other folks who are also working on it, I would imagine, but I don't know of any specific studies that have shown that yet. But I think that that is probably something that most people speculate.
Got it. Thank you.
Okay, Dimon, you want to go next?
Hi. Thank you for the very inspiring talk. I really liked it. So my question is that the glycan gating mechanism that you showed is kind of observed using the weighted ensemble trajectories. So my point is that I would expect the trajectories which show the transition are the ones which will have like very low weights or low probabilities. So is it possible that there are some alternative, like a high probability but slower pathways that are kind of missed in this approach?
I guess it's a good question, but so, you know, you run so many of these, so you get like, you really do get, I mean, it's like thousands of trajectories that you run. So you really do sort of get, I think, a pretty good sense for what's happening mechanistically. And we see, you know, I can tell you that when you analyze all these independent pathways, you really do sort of see a conservation of these particular behaviors. And they are independently, in some sense, independently generated. You could show that they're sort of like not correlated with each other. So I don't know. I mean, I think that's also, I mean, it's possible, I guess maybe it's possible, but I think this does a pretty good, I think that approach in general does a pretty good job of finding the key mechanistic drivers, you know, for these different biological processes. And then of course, the fact that we, you know, have the experimental backup doesn't hurt. There could be other things that we're missing. You know, the other thing that we struggle with, which I would have loved to get, would be kinetics. But we haven't been able to really sample enough yet to actually resolve the timescales of motion directly from the MD. But that's something that I was sort of hoping we might get in future work, but it's still TBD.
Yeah, that would be really interesting. Yeah. Thank you.
Welcome.
Okay, so 2 more questions and then I'm going to close it because everybody's busy. Chun. All right, I don't hear anything. Chun?
Yeah, I just unmute myself. Thanks
ahead.
for the talk. It's very interesting. So I remember that in the talk, one of the emphasized about how much the sugar can do for the spike protein in protecting it. And of course, there are people like saying that maybe you can use different sugar. Have you thought of like maybe running like a cross-grain simulation or like even a docking for like a virtual screening of all existing antibodies, since we have vaccine now, and see how the sugar may like the different use of sugar may affect like the affinity to their binding target or even like defecting them, or like whether some new antibody can do that fit particular set of sugar? Yeah.
So I will tell you that yes, and this sort of ties into what Zan was saying too, that you know, there is this thing, there's like heterogeneity to the sugars, to the glycans that are on the molecules. It could go, even in like the same expression system, there's heterogeneity, but then person to person to person, there's also heterogeneity, like she was saying, depending on all sorts of things. The best work that I've seen to try to disentangle the exact effect of particular glycans and glycoprofiles is actually being done by Elisa Fada, who I mentioned. And also Rob Woods, I think, has some very nice work. Well, where they change the type of glycans at a particular site, and then they see the extent to which the shielding is there or that it's affecting dynamical motion and so forth. We haven't, for us, you know, I have to tell you, these projects in SARS-CoV-2 have really become, it's like a triage situation with, you know, people, experimentalists like asking for particular things, and we only have so much bandwidth. So we haven't actually been able to go through really carefully and do that, but others are working on that, and I would steer you to their studies. So I would look to see what Elisa Fada is doing. And I'll say, you know, one of the things that she has actually really interesting in a preprint in the bioarchive is how there's a change in glycosylation actually potentially involved in the jump from bats or pangolins to people. So from like SARS-1 to SARS-2, there's some interesting changes there. So that has all types of effects for antibody bindings, for evolution, etc.
Okay, good. Can you show our picture in San Francisco one more time, Romy? I have a little story to tell everyone. I got my green card on that day, and ironically, I was wearing all green And then actually we had Margarita celebrating that. That was funny.
That was so great.
Well, thank you very much, Rommie. This was a fantastic talk and seminar and conversation. We really enjoyed that. And thanks for emphasizing how much we can learn from these simulations rather than just showing that we can do it. So it was really inspiring. Thanks again for being with us. We really enjoyed that. So I understand that you're meeting with Jale after this, but so the seminar is over. Thanks for everyone for being
Thank
here.
you for having me.
But some friends can stay on if you want to continue chatting. I hope Jale doesn't mind.
Yeah, I'm on. I can stay on. And
Martin,
thanks for
One thing especially for the students, Rommie did a really nice job of, talking about Klaus and the inspiration you gave. But I talk often about you to my students, how this one phrase I learned from you is when you were my graduate student, you have to take possession of the project. And, I never had to ask you to come in or come forth with new results. It was like, when are you going to meet with me, Zan? I have great new things to tell you.
Quit avoiding me. Don't make me come to your house.
Exactly.
But if you went to their house, they'd feed you. So it wasn't.
I know. I like going to the house. I like
Okay,
going to the house.
enough. Turn it over to other students. We're great, Rommie. Bye-bye.
Suzanne. Bye.
Good to see you. I mean, I was just telling Elizabeth, it feels like we are back in San Francisco.